Hi, DrBiology here, and this is Essential Math Skills for A-Level Biology, and I'm going to be looking at chi-squared today, which is a statistical test. So first things first, here we go. So here is the chi-squared formula. Now luckily you don't need to learn that, okay? I'm going to show it you and I'm going to go through it, but it's really important to state this, that there are a few basics, particularly from your exam board AQA. So in written examinations you will not be asked to perform a calculation using a statistical test. It will be important for you to understand how to select a statistical test that is appropriate for given data and to be able to interpret the results of such a statistical test. You could also be asked to explain your choices and interpretation. Okay, but I think you do need to have an example, so I'm going to give you an example today of how you would work out the chi-squared, but then we're going to spend a bit of time about how you interpret the results, which is very important for your A-level biology. So first of all, chi-squared test. So what is it? Well, it compares measurements relating to the frequency of individuals in defined categories. So chi-squared is used to test if the observed frequency, in other words, the observed observations through trapping, for example, if you're trapping small insects, or if you're doing some field work where you're counting things, and it fits the frequency you expected or predicted. And I'm going to use the example of periwinkles. Now, periwinkles like to live in what we call the splash zone. So the splash zone of a rocky shore zonation. Okay, so obviously there are different animals and different seaweeds. You can see all the different types of seaweed, and periwinkles like to nibble on the seaweed. So this is going to be a little example of where we're going to look at the frequency of periwinkles and in different types of seaweed. So here we go. So they eat seaweed. Do they prefer certain species of seaweed? So how would we do a study to find that? Well, what you could do is you could count the number of periwinkles, so that's the frequency, on different species of seaweed. So in this case, these are going to be the categories. Now, the first thing you need to do in a chi-squared test is have a null hypothesis. So null hypothesis is where there is no significant difference between the observed and expected frequencies. So in terms of our example here, we're looking at that there is no significant difference in the type of seaweed the periwinkle prefers. So here's a table of data. So someone would have gone out and basically counted the number of periwinkles living on a type of seaweed. They probably would have done a random, I would imagine, a random use of random quadrats to reduce bias. But you can see the count there of periwinkles, and there seems to be a lot on serrated and bladderwrack, less on egg and spiral and other seaweed, but we can't give a statistical observation of that data. So what we can do is use chi-squared. So what you can see is the observed frequencies, so that is the actual observation frequency. The expected frequency is the equal number, so there was 100 divided by the number of categories, which is 5, so you'd expect to see 20 periwinkles on each piece of seaweed. Then observed minus expected, and then observed minus expected squared, and then divided by expected. And that would give us the sum of, and that would give us the chi-squared total of 79, which is quite a high figure. So what in fact does that mean? What does that mean about the data? And that's what we're going to look at now. So the first thing we need to do is work out what we call critical value and degrees of freedom. So what we're going to need to do is look at a table of critical values. Now, in an exam, they won't give you these critical values, but you do need to understand what it means. So the critical value represents the borderline between accepting or rejecting our null hypothesis. So we get the critical value from a data sheet, this table sheet, but it depends on what we call the number of degrees of freedom. So degrees of freedom is the number of categories minus 1. So in this case, there were 5 different seaweed categories, so different species of seaweed. So it would be 5 minus 1, which is 4 degrees of freedom. We look at 4 degrees of freedom, and the critical value is 9.49, and that's what we're going to compare our chi-squared value to. Now, before we go on to compare them, we need to also know about the p-value. So p-value stands for probability value, so it indicates how likely it is that result occurred by chance alone. So if the p-value is small, it indicates the result was unlikely to have occurred by chance alone, so therefore we could say they are statistically significant. Now, in terms of probabilities, in terms of writing them as a number, 0 would mean something is pretty impossible, whereas 1 means something is pretty certain. So if we have p equaling 0.05, that means something is likely to happen 5 times in 100. So we're pretty certain that there's a difference, there is a statistical difference in the data. So to test our hypothesis, we're going to need to choose a probability level. Now again, in an exam, they will usually show these 2 probabilities, okay, one or the other. So in science, these are always very small, and they show that the results are probably not due to chance. And I say probably because there is still a chance that it is chance. Okay, so the first one, p is smaller than or equal to 0.05. So it means 5% of the time a significant result will be due to chance. Or we could think of it another way, that basically there's less than 5% that it is due to chance. And then p is smaller than or equal to 0.01. So 1% of the time a significant result will be due to chance. So let's interpret the results that we've got. So just a reminder, our chi-squared was 79, our critical value is 9.49. So chi-squared gives a number which indicates how big the difference is between the observed and expected data. Okay, if the chi-squared value is small, then there's a small difference between the observed and expected. So this means the null hypothesis is accepted or likely to be correct. In other words, the snails don't mind which seaweed they live on, but we found there was a big difference. So the value was huge. There was a huge difference between the observed and expected data. So this means the null hypothesis can be rejected. So we're saying the snails do indeed have a preference for living on a particular seaweed. So we can see that the critical value of chi-squared at 5% significance and 4 degrees of freedom is 9.49. So our calculated value is 79. So it's much bigger than that critical value. Okay, so this means we reject the null hypothesis, and we're saying that snails are not scattered around randomly, and it infers that they do seem to prefer to live on a certain species. Now, how would you state that in an exam? We would show it like this. So the value of chi-squared, and you could say what it is, 79, is greater than the critical value of 9.49. So there is less than 5% probability, because we're doing it at p is smaller than or equal to 0.05, that the difference between the observed and expected data are due to chance. So therefore we reject our null hypothesis. Okay, so what we're going to do now is look at some actual exam questions to see how do you interpret the data. So here is our first example. So it says, in an investigation, cyanogenic and acyanogenic plants were grown together in pots. So cyanogenic just means they're quite poisonous to things like slugs. So slugs were placed in each pot, and records were kept of the number of leaves damaged by the feeding of slugs over a period of 7 days, and they show you the results of undamaged and damaged. Okay, and then they said a chi-squared test was carried out, suggests the null hypothesis that was tested. So what I suggest you do is pause and have a go at these questions. So the null hypothesis is when the observed and expected there is no difference. So what we can say is there was no significant difference in damage between cyanogenic and acyanogenic plants. So when you give the null hypothesis, use the example that they've given you in the question. Then it says chi-squared was calculated. So when this value was looked up in a table, so that would be a critical value table, it was found to correspond to a probability of less than 0.05. So that's a significant difference. So what conclusion can you draw from this? Well, we can conclude that the difference is significant. Okay, it's not due to chance. We therefore reject the null hypothesis, and therefore being cyanogenic does help protect from slug damage. Notice that these kind of questions, 3 marks, they want you to say about the difference being significant. They want you to say that you reject or accept a null hypothesis, and then use the information that is there to help answer the question. Okay, second question here. So again, let's have a look at it. So this is ecology based, about orange star lichen on the walls of a building. So they recorded the number of colonies within a 50 centimetre squared quadrat. Chi-squared test was applied to the results, okay, and they looked at 3 walls. Sorry, that's the most important thing, the north, south and west facing walls. So again, a very similar type of question, give a null hypothesis. So basically you're thinking about, well, there's no difference in the number of lichens growing on walls that face in different directions. So complete the following table. Okay, well, you need to add up the north, south and west, and if you add them up and divide by 3, you'll then get the expected, which in this case is 36. How many degrees of freedom were in this chi-squared? Well, there are 1, 2, 3 categories. So remember, degrees of freedom is 3 minus 1. So in this case, it will be 2. And then a chi-squared value of 15.5 was calculated as a probability of less than 0.001, which is highly significant. Explain what this means. Well, we need to talk about significance. Well, yes, there is a significant difference. We can reject the null hypothesis. Okay, we know that the difference is not due to chance, and the direction of the wall, the faces, does have an effect on the population of lichens. Okay, so I hope you found that very useful. There's going to be more videos on essential math skills for A-level biology. Please do put your bell alert reminder on for all my new videos. I'll see you soon.